The purpose of this presentation is to provide further insight into the Questions and Answers document developed by the ICH Q11 Implementation Working Group on the selection and justification of starting materials and finalized in August 2017. This presentation is protected by copyright and may be used, reproduced, incorporated into other works, adapted, modified, translated, or distributed under a public license, provided that ICH's copyright in the presentation is acknowledged at all times. The presentation is organized in the form of background information on the ICH Q11 Parent Guideline, elaboration of the ICH Q11 General Principles from Section 5.1 in the Parent Guideline, and 2 case studies which have been developed to illustrate specific aspects of the guidance. It is recommended that the viewer should be familiar with both the ICH Q11 Parent Guideline and the ICH Q11 Questions and Answers document in order to benefit from the material in this presentation. This presentation will also make reference to other ICH guidelines, and the viewer should also make reference to those documents in order to understand the concepts described in this presentation. The ICH Q11 Parent Guideline provides guidance to industry and regulators concerning the development and manufacture of drug substances. The scope of ICH Q11 covers chemical substances and substances of biological and biotechnological origin. It covers both novel substances and existing drug substances, and it addresses both traditional and enhanced approaches to drug substance development. This guideline was adopted by the ICH Management Committee in May 2012. The ICH Q11 Questions and Answers document has been developed specifically to provide further guidance on the selection and justification of starting materials, as described in Section 5 of Q11. Section 5.1 is organized as General Principles for Selection of Starting Materials for Synthetic, Semi-synthetic, and Biological and Biotechnological Substances. Section 5.2 describes the information that should be considered by way of justification for the selection of a particular starting material or materials. The Q11 Questions and Answers document provides further clarification on how to apply the general principles in the Parent Guideline based on science and knowledge of the proposed manufacturing process and its controls. It contains 16 questions and answers, providing clarification and promoting convergence on the concepts for regulators and industry. It is intended to be used as support for and not instead of the Parent Guideline. It also contains a useful decision tree that has been developed to promote understanding of the application of the general principles. It is important that all of the ICH Q11 general principles are considered in the selection and justification of proposed starting materials, rather than selecting just a few principles in order to justify particular starting materials. If the proposed starting material does not meet all of the general principles, the acceptability of that starting material should be further explained. The scope of the Q11 Questions and Answers document follows that of the Q11 Parent Guideline. Its focus is on synthetic drug substances, and it was not written to take into account the specific considerations required for biological and biotechnological substances. It is important to be aware that both the Parent Guideline and the Questions and Answers document are intended to apply to applications for marketing authorizations. They are not intended to apply directly to the clinical development phases of the product life cycle. The guidance is also applicable to master files. It is anticipated that the information submitted in master files will comply with the Parent Guideline and the Questions and Answers document. The guidance in the Questions and Answers document is intended to be consistent with and to support the Parent Guideline. It is not intended to be applied retrospectively to marketing authorizations or master files that have already been approved. However, the guidance should be taken into account if significant changes are proposed to the drug substance manufacturing process or its controls. It is important to recognize that a starting material accepted for one manufacturer's synthesis may not be acceptable for a different manufacturer's synthesis unless it complies with the guidance. This slide summarizes the general principles for the selection of starting materials as described in ICH Q11. The full text for these general principles can be found in Appendix 1 to this training material. It is important to note that a manufacturer should consider all of the general principles in the selection and justification of starting materials, rather than to focus on a specific principle or a select subset of principles. If a proposed starting material does not meet all of the general principles, a rationale should be included in the application explaining why the starting material is considered to be appropriate. It should be recognized that the general principles are based on the assumption of understanding of the manufacturing process and of the formation, fate, and purge of impurities in the drug substance. It is expected that this information will be included in the regulatory file. The formation, fate, and purge of mutagenic impurities are important considerations for synthetic drug substances. Manufacturing process development studies, for example, pilot-scale operations, should provide knowledge of the operating conditions necessary to assure the quality of the drug substance. The next slides are intended to illustrate the considerations that should be taken into account in order to address the ICH Q11 general principle that enough of the drug substance manufacturing process should be described in the application for regulatory authorities to understand how impurities are formed in the process, how changes in the process could affect the formation, fate, and purge of impurities, and why the proposed control strategy is suitable for the drug substance manufacturing process. It is recommended that the manufacturer should identify those steps in the manufacturing process that impact the drug substance impurity profile. The manufacturing steps that are the source of impurities that impact the drug substance impurity profile should generally be included in the description of the manufacturing process in Section S2.2 of the application or in the master file. The exception to this situation is the case of impurities that persist. The explanation of impurities that persist, as used in Q11, will be given later in this presentation. After the manufacturing steps that are the origin of impurities that impact the impurity profile have been identified, the applicant should then look at the steps immediately preceding these in the process. Those manufacturing steps that need to be carefully controlled to prevent the generation of impurities that would otherwise impact the drug substance impurity profile should also be included in Section S2.2. For example, if the process parameters need to be tightly controlled or a manufacturing step includes a unit operation to remove specific impurities that would otherwise carry over into the drug substance. If, after having conducted this exercise, the potential starting materials are a small number of chemical transformation steps from the final drug substance, the starting materials should be defined one or more manufacturing steps further upstream of this point. This is in order to ensure that the process is sufficiently controlled under GMP conditions. In considering how many more manufacturing steps to include in S2.2, an applicant may propose a control strategy, including comprehensive analytical methods, for example, those based on different physical and chemical separation and detection principles, in order to mitigate this risk. The threshold for determining when impurities impact the drug substance impurity profile depends on the toxicity of those impurities. For non-mutagenic impurities, the threshold is the identification limit for impurities as defined in ICH Q3A. For mutagenic impurities, 30% of the ICH M7 acceptable limit serves as the threshold. If an impurity in the drug substance exceeds these levels, the manufacturing step responsible for the generation of that impurity should be identified. This is a manufacturing step that impacts the drug substance impurity profile. The impurity in the final drug substance may have been transformed in the manufacturing process. A step that impacts the impurity profile is the source of the precursor to that impurity. An exception to the inclusion of manufacturing steps that impact the drug substance impurity profile is in the case of those impurities that originate in early manufacturing steps and persist or are carried over into the drug substance. If the impurity in the drug substance or its precursor is carried over other manufacturing steps that do not in themselves impact the impurity profile, this can be considered to be an impurity that persists. In this case, the manufacturing step that is responsible for the impurity that persists does not need to be included in Section S 2.2. This concept is illustrated in the subsequent examples. This slide illustrates Example 4 from the Q11 Parent Guideline. It is recommended that the viewer should review Example 4 in the appendix to ICH Q11 Guideline. In the example, an enantiomeric impurity is generated in Step 1 of the manufacturing process and is the source of an impurity that occurs in the drug substance in excess of the identification threshold. This impurity impacts the impurity profile of the drug substance and is also an example of an impurity that persists over multiple steps, in this case, Steps 2 and 3. The other impurities that impact the impurity profile of the drug substance originate in manufacturing Steps 4, 5, and 6 of the process. It is important to note that the concept of impurities that persist is not limited to chiral impurities. According to the Q11 general principles, the applicant should initially consider compound A to be a potential starting material for the drug substance, since impurity 1 originates in Step 1 and impacts the impurity profile of the drug substance. However, Steps 2 and 3 of the manufacturing process do not introduce impurities that impact the impurity profile of the drug substance, nor do they require careful control within the control strategy. The enantiomeric impurity, Impurity 1, is an example of an impurity that persists, as described in the question and answer 5.8. In this case, the applicant may propose to control the carryover of the enantiomeric impurity in the drug substance within the control strategy. In this case, D may be accepted as the starting material. In this slide, Example 4 has been modified to illustrate a different situation in which the impurities that impact the impurity profile of the drug substance originate in Steps 1, 3, and 5. In this example, the enantiomeric impurity is still considered to be an impurity that persists. However, since impurity 2 is generated in manufacturing Step 3, D is no longer a suitable starting material, and the starting material should be defined upstream of D, for example, C. In this slide, the example has been modified again to illustrate a situation where the impurities are formed in manufacturing Steps 1, 2, and 3 of the process. In this situation, Impurity 1 is no longer an impurity that persists, since Step 2 and 3 of the process also contribute to the impurity profile of the drug substance, and A would be considered an appropriate starting material. Another important aspect in considering manufacturing steps that impact the drug substance impurity profile is the origin of mutagenic impurities in the drug substance. The Q11 questions and answers have clarified that for the purpose of defining the starting material in the regulatory file, mutagenic impurities that carry over into the drug substance in excess of 30% of the ICH M7 acceptable limit are considered to impact the impurity profile. The manufacturing steps in the process which generate these impurities are steps that impact the impurity profile. The exception for persistent impurities described in the previous examples is also applicable for mutagenic impurities. In order to identify potential mutagenic impurities in the manufacturing process, it is recommended that the applicant should use the ICH M7 hazard assessment elements to determine the risk of mutagenic impurity carryover in the drug substance. This should include evaluation of impurities that carry over in the drug substance and evaluation of reagents and intermediates used in the drug substance manufacturing process from commercially available raw materials. Note that this may require evaluation of steps upstream from the proposed starting material in the regulatory file. Reagents and intermediates should be assessed for mutagenicity if they are likely to occur in excess of 30% of the ICH M7 acceptable limit in the drug substance. In the case of the evaluation of the mutagenicity of impurities in commercially available raw materials or impurities in intermediates, the manufacturer may use risk-based reasoning to evaluate the risk of carryover in the drug substance in excess of 30% of their acceptable daily intake. In those cases where the drug substance is itself genotoxic and intended for treatment of advanced cancer indications, mutagenic impurities are not considered to impact the impurity profile unless they occur in excess of the ICH Q3A identification threshold. The concept of impurities that persist is also applicable to mutagenic impurities for the purpose of selecting the starting materials for a manufacturing process. The ICH Q11 general principles do not recommend that all steps involving mutagenic reagents or impurities are included in Section S 2.2 of the dossier. The General Principles and Questions and Answers document describe the considerations that should be taken into account in defining starting materials with reference to risk of mutagenic impurity carryover. Once the starting materials for a manufacturing process have been defined according to the Q11 general principles, any changes to the process that impact the risk of carryover of mutagenic impurities should consider the guidance in ICH M7. The Q11 Questions and Answers document provides further information on the distinction between commercially available chemicals and custom synthesized chemicals. The parent guideline clarifies that commercially available chemicals are suitable starting materials, and it is not necessary to provide justification of their selection in accordance with the general principles. Manufacturers have sometimes proposed starting materials that do not meet the intended definition of commercially available starting materials. This slide clarifies that commercially available starting materials are chemicals sold as commodity materials in non-pharmaceutical markets. A chemical material made in-house specifically to a drug substance manufacturer's requirement does not meet the definition of a commercially available chemical. Similarly, a chemical made externally from the drug substance manufacturer, but according to the manufacturer's requirement, does not meet the definition of a commercially available chemical. A chemical available for purchase, but where its only use is for manufacture of pharmaceuticals, also does not meet the definition of a commercially available chemical. If the proposed starting material is not within the definition of a commercially available chemical, the suitability of that starting material should be justified with reference to all of the general principles, or another suitable starting material should be proposed. The following slides contain 2 case studies which exemplify many of the Q11 general principles and concepts discussed so far. There isn't any intent to suggest that a particular number of synthetic steps would be considered enough or not, nor are they intended to prescribe a particular way of presenting data or justification. Case Study 1 is based on a real example, although the structures have been replaced with letters to protect confidentiality. The scheme shows a convergent synthetic process starting with a range of raw material inputs. In this case, the applicant proposed 3 starting materials, A, B, and G, highlighted in blue. Intermediates in the process are indicated by yellow boxes. Starting materials A and B are introduced early in the process, and there are multiple reaction steps and isolations until the final intermediate, F, is attained. In the final step, intermediate F is coupled with the 3rd proposed starting material, G. G itself is prepared in multiple chemical transformations from compounds O and M. Step 8 consists of a resolution of racemic M to afford enantiomerically enriched L. This table summarizes the impurities potentially present in the drug substance, how they are controlled, and where they originate from. It is used as a way of summarizing the information relevant to the case study. It is not intended to convey that applicants should create such a table. Impurity 1 originates in Step 6, the conversion of O to N. It is a mutagenic impurity which is difficult to purge and persists across multiple steps all the way to the drug substance. The applicant proposed to control it to no more than the ICH M7 acceptable limit in intermediate G rather than the drug substance. The other impurities are all non-mutagenic and are specified in the drug substance above the ICH Q3A identification threshold. Impurities 2, 4, 8, and 9 all originate in the final 2 steps of the process and don't originate from impurities in any of the proposed starting materials. Impurity 3 originates 4 steps upstream of starting material A. Impurities 5 and 6 originate 2 steps upstream of starting material B. All of these impurities carry over to the active substance above the identification threshold and are impurities that persist. Impurity 7 is the minor enantiomer of the drug substance and originates in the Step 8 resolution. It is also an impurity that persists and is specified in proposed starting material G. This slide goes into more detail on the considerations for control of mutagenic impurity 1 and the impact on the selection of the proposed starting material. The applicant conducted a risk assessment to determine which raw materials, intermediates, and impurities were mutagenic in line with ICH M7. Impurity 1 was determined to be mutagenic. Without additional controls, Impurity 1 was found to be present in the drug substance at levels above 30% ICH M7 acceptable limit. Impurity 1 is therefore considered to impact the impurity profile, as defined in Question and Answer 5.7. Impurity 1 originates in Step 6 and carries across 4 subsequent steps into the drug substance. Since no further impacting impurities are formed in Step 7, then Impurity 1 is a mutagenic impurity that persists. In line with Question and Answer 5.10, it is not necessary to include all steps involving mutagenic impurities in the process description. Accordingly, the applicant chose to control the level of impurity 1 by limiting its precursor in compound G with an acceptance criterion of not more than the ICH M7 acceptable limit, i.e., in line with option 2 of that guideline. The following 2 slides show how the decision tree from Annex 1 of the question and answer document can be used to help decide which of the proposed starting materials are acceptable. Each box has a cross-reference back to the relevant question and answer for the concept under consideration. The first part of the decision tree evaluates the structural elements of proposed starting materials A, B, and G. Since the structures have been removed from the synthetic scheme, it's not possible to discuss this part in detail in this presentation. However, the general principles can still be discussed. The first decision box asks whether a given starting material has defined chemical properties and structure. Starting materials A, B, and G all meet this principle, as demonstrated by information provided in the dossier. The second box asks whether a given starting material is incorporated as a significant structural fragment into the structure of the drug substance. Again, all 3 starting materials meet this principle. The majority of each starting material is included in the final structure of the drug substance. The third box asks whether a given starting material is commercially available according to the definition given in ICH Q11 and clarified in Question and Answer 5.6, i.e., one that is sold as a commodity in a pre-existing non-pharmaceutical market in addition to its proposed use as a starting material. None of the proposed starting materials A, B, and G could be demonstrated to meet this criterion by the applicant. Given that the answer to this final question is no, then Part 2 of the decision tree should be consulted. The second part of the decision tree considers impurities which impact the drug substance impurity profile, and whether enough of the manufacturing process has been included in the dossier. The first decision box asks whether steps upstream of the proposed starting materials impact the impurity profile. It was demonstrated that impurities generated in steps upstream of all the proposed starting materials impact the drug substance impurity profile, as shown in the table on slide 22. However, it was also shown that these can all be considered as impurities that persist. The next factor to consider is whether steps immediately upstream of each proposed starting material need to be carefully controlled to prevent generation of impurities which would otherwise impact the drug substance impurity profile, or whether the upstream steps include unit operations added to control impurities. For example, adding purification steps to remove impurities which would otherwise have an impact in order to avoid defining an earlier compound as a starting material would not be acceptable. Based on the nature of the reaction steps, the answer to this question for all 3 impurities was no. The final consideration is whether only a small number of chemical transformations are included between each proposed starting material and the drug substance. There are 5 chemical transformations between both starting materials A and B and the drug substance. However, proposed starting material G is only 1 step away. Therefore, the applicant was requested to redefine the starting material to an earlier point in the synthetic route. This is to help mitigate risks associated with future changes to the synthetic route or supplier of G, and potential contamination arising from non-GMP manufacture. These risks include the presence of impurities or contaminants, which may not be detected by the analytical methods used for release testing. Performing additional steps under GMP and describing them in the dossier helps ensure that the control strategy remains effective and that the quality of the active substance remains adequate throughout the life cycle of the product. The applicant could also have considered other approaches to risk mitigation in determining which upstream compound to select as a starting material. For example, inclusion of analytical methodologies designed to detect a wide range of possible impurities. This slide shows the revised synthetic route following redefinition of originally proposed starting material G to I and J. Compounds H and G are now considered intermediates. There are no impurities generated in steps 7, 9, and 10, which impact the impurity profile. Impurities generated in steps 6 and 8 impact the drug substance impurity profile because they are not readily purged in the downstream process and are therefore considered to be impurities that persist. As a result, there was no need to define O and M as starting materials, and I and J were considered acceptable. There are now 3 steps between starting materials I and J and the drug substance, which was considered enough in this case. However, to reiterate the points made on slide 20, this does not mean that 3 steps will always be considered enough, nor that at least 3 steps are mandatory between a given starting material and drug substance. The decision must be made on a case-by-case basis and depends on the nature of the chemistry being carried out, the overall synthetic route, and the principles explained in Question and Answer 5.11. Case Study 2 is based on the Securamill mock dossier found at the link on the slide. However, the reader should note that the synthesis used for Securamill has been significantly modified in structures, impurities, reactions, and controls for the purpose of these training slides in order to exemplify specific aspects of the ICH Q11 Q&A. This case study is not intended to be read in conjunction with the Securamill mock dossier, and no inferences are intended. This slide illustrates a synthetic route to manufacture a drug substance, which is designated as C9. Interesting features about this synthetic route include a process that consists of 6 covalent bond-forming steps, and the drug substance is a free base, not a salt. 2 of the intermediates, C3 and C5, are not isolated as indicated by the brackets in the scheme. The drug substance contains 2 chiral centers. One chiral center originates from the beta-aminonitrile, compound C2, which is purchased. The second center is formed in step 4 via a highly stereoselective cyclization reaction to make compound C6. The second chiral center is introduced diastereoselectively in step 4 under substrate control. The other important thing to note is that both the methyl and ethyl chloroformate used in steps 3 and 5 in this example are considered reagents, since neither makes up a significant structural fragment of the API, and both are common reagents. This table summarizes data on non-mutagenic impurities in the drug substance that exceed the ICH Q3A identification threshold and mutagenic impurities that may be introduced in a proposed starting material or later in the process. C9D1, which will be illustrated on the next slide, is a diastereomer of the drug substance and indicated with the nomenclature of D1. All other non-mutagenic impurities in the process are controlled below the limit for unspecified impurities of 0.10%. C3, C4, C5, C6, and C8 are mutagenic impurities. The threshold of toxicological concern for these impurities is established at 25 ppm. Remember that mutagenic impurities are considered to impact the impurity profile when they are likely to be present in the drug substance above 30% of the ICH M7 acceptable limit. For these impurities, the ICH M7 acceptable limits are equal to the threshold of toxicological concern. Both C6 and C8 are specified at the threshold of toxicological concern level, and they impact the drug substance impurity profile. C3, C4, and C5 are present at a level of less than 30% of the threshold of toxicological concern in the drug substance and therefore do not impact its impurity profile. The structure of C9D1 is the diastereomer with opposite configuration at benzylic nitrogen and is formed during the step 4 cyclization. C9D1 is a non-mutagenic impurity and is specified in the drug substance specification at 1.0%, that is, above the identification threshold. Therefore, this impurity impacts the drug substance impurity profile, and step 4, the process where the impurity originates, is a step that impacts the drug substance. C6 is an intermediate produced in step 4 of the synthetic route and a mutagenic impurity that occurs in drug substance in excess of 30% of the ICH M7 acceptable limit. C8, benzyl bromide, is a commercially available commodity in non-pharmaceutical markets. It is a significant structural fragment, not a reagent, and it is introduced in the last step of the synthesis. Therefore, C8 is acceptable as a starting material. Impurities in benzyl bromide do not impact the drug substance impurity profile. If benzyl bromide did contain impurities which needed to be removed through an additional purification operation by the manufacturer prior to its use to ensure the quality of the drug substance, then any purification operations should be described in Section S2.2 and performed under GMP, see ICH Q11 5.2.1 and Question and Answer 5.14. This should also include appropriate specifications for pre-purified incoming material and purified material. Benzyl bromide would be considered acceptable as a starting material since it is commercially available. However, basic information should be provided on the starting material: chemical name, chemical formula, and molecular weight, information on the impurity profile of the starting material, and how the control strategy for the drug substance manufacturing process justifies the starting material specification. See Q5.14. Both C3 and C5 are non-commercially available chemicals or custom chemicals, and therefore should be assessed according to the ICH Q11 general principles and Q11 question and answer clarification. However, non-isolated intermediates are usually not considered appropriate starting materials. Would C7 be an acceptable starting material? It is the last chemical intermediate in the manufacturing process, and therefore multiple transformation steps would not be described if this were selected as a starting material. In addition, there are steps upstream of C7 that generate impurities which impact the drug substance impurity profile, most notably Step 4. The proposal of C7 would not result in enough of the manufacturing process under GMP, as described in Question and Answer 5.11. Therefore, C7 would not be acceptable as a starting material. Would C6 be an acceptable starting material? Just like C7, there are steps upstream of C6 that generate impurities which impact the impurity profile. It should be noted that Step 4 is also an important step that is controlled in order to ensure the required stereoselectivity at the benzylic nitrogen. The proposal of C6 would potentially not result in enough of the manufacturing process under GMP, as described in Question and Answer 5.11. Therefore, C6 would not be acceptable as a starting material. Would C4 be an acceptable starting material? Impurities that impact the drug substance impurity profile do not originate in or upstream of this intermediate. The enantiomer is controlled in C2 in accordance with the vendor specification, and FATE and Purge studies show that it is purged to well below the ICH Q3A identification threshold in the drug substance. In addition, steps upstream of C4 do not require careful control in order to assure the quality of this chemical, nor do they include unit operations specifically added to ensure adequate purity. See Question and Answer 5.11. The proposal of C4 provides multiple chemical transformation steps to be included in Section S2.2. Therefore, C4 would be acceptable as a starting material. It should also be noted that if the enantiomer of this material did carry through to the drug substance at an impactful level, this impurity could be considered an impurity that persists across multiple steps. See Question and Answer 5.8. So C4 could still be acceptable, provided that it was justified in accordance with the other Q11 principles. This slide illustrates the conclusion of Case Study 2, where the two starting materials displayed in the red boxes were accepted. C8 is a commercially available starting material. C4 is a custom starting material that complies with the ICH Q11 general principles for determining starting materials as outlined in the Question and Answer document. The purpose of this slide deck is to support the selection of appropriate starting materials for the synthetic chemical manufacturing process described in Section S2.2 of the Common Technical Document. The selection of suitable starting materials is based on scientific data and process knowledge and understanding. It is recommended that all of the relevant guidance in ICH Q11 and the supporting questions and answers should be considered when proposing a starting material or materials in the application. This concludes our presentation. We hope you found it useful. Thank you for watching.